Yes, in the previous class you are studying the genetic code. So, genetic code means what I have explained. So, genetic code means here on mRNA means it is what? Messenger RNA. The genetic information is in what? Depicted. Genetic information is depicted in the form of the code. And yesterday you also studied there are three systems of here the coding is there. That is one is called singlet codon, and by this it is possible to what give the what codes to 4 codes only to the what the amino acids. There are 20 amino acids are there, but only 4 code A, U, G, C, and which do not fulfill to give codes to all the 20 amino acids, correct? So that is why this was failed. Then the next one it was singlet codon system. Sorry, doublet codon system. Singlet is over. Doublet codon system, and here in the doublet codon system I have written here AUGC and AUGC. When it is crossed like this by doing checkerboard system, and we will get totally the 16 code. Again, is not possible to give the codes to all the 20 amino acids, because once again 4 will remain as a the separate. Only 16 amino acids only we can name by the codes. So that is why this was also what failed. And the last one, and that was a triplet codon system. Triplet codon system, and a triplet codon system in which here what the totally 64 codon we will get. And by this it is possible to give the code to all the 20 amino acid, and this was first time discovered by what Marshall Nirenberg and Hargobind Khurana of India, who is a citizen of Canada, and who first time gave the explanation regarding the genetic code, and that to be depicted on the mRNA, and that to be always in the ಟ್ರಿಪ್ಲೆಟ್ ಕೋಡಾನ್ ಸಿಸ್ಟಮ್. ಟ್ರಿಪ್ಲೆಟ್ ಕೋಡಾನ್ ಸಿಸ್ಟಮ್ ಅಂದರೆ 3 ಏನು ಸೀಕ್ವೆನ್ಸ್ ಕೂಡಿ ಒಂದು ಕೋಡಾನು, ಅದಕ್ಕೆ ಟ್ರಿಪ್ಲೆಟ್ ಕೋಡಾನ್ ಸಿಸ್ಟಮ್ ಅಂತ ಕರೀತಾರೆ. ಆ್ಯಂಡ್ ದಿಸ್ ಈಸ್ ದ ರಿಯಾಲಿಟಿ ಇನ್ ಆಲ್ ದ ಲಿವಿಂಗ್ ಆರ್ಗ್ಯಾನಿಸಮ್ ಫ್ರಮ್ ಪ್ರೊಕ್ಯಾರಿಯೋಟ್ ಟು ಮಲ್ಟಿಸೆಲ್ಯುಲರ್ highly organized plant and animal. Same type what here codon system is there, and that to be depicted in the mRNA, because mRNA that contain genetic message, and that is what triplet codon system. And here now today you have to study the how triplet codon systems are generated or made, and that you have to study. See, now in this triplet codon system, 1 minute, I have to make a column. Already that is you studied in the online classes of your Zoom class, and where you have given the notes also. Notes ಕೂಡ ಅಲ್ಲಿ ಕೊಟ್ಟಿದ್ದೇನೆ. How you have to do the genetic codes ಅನ್ನು ಹೇಗೆ ಅದನ್ನು ಮಾಡ್ಕೊಳ್ಬೇಕು ಅಂತ ಹೇಳಿದ್ರೆ, and that is to be here. See, columns you have to do like this. Like this you have to do the columns. See, like this you have to make a column, and then you have to write the here the nitrogen bases UCAG like this, and then UCAG. So like this you have to write on both the side, and then one more column you have to extend here. Here UCAG, same UCAG. See, triplet codon system is 3 side. We have to now prepare the codons. So how you have to write C. Next UA and then UG. ಅಂದರೆ ಇಲ್ಲಿ ಕಂಪ್ಲೀಟ್ ಬರಬೇಕು. UCAG ಲಾಸ್ಟ್ ಸೇಮ್ UCAG ಎಲ್ಲ ಕಡೆ ಹಾಗೆ UCAG. ನೆಕ್ಸ್ಟ್ UCU. ನೆಕ್ಸ್ಟ್ UCU. ನೆಕ್ಸ್ಟ್ UC what C. Next UCA and then UCG. This is also last UCAG. You see. Then next one is UAU, UAC, UAA, UAG. Last. Similarly, UG, UGU, UGC, UGA and UGG. Correct. Then next column is CU, CU, CUU, CUC, CUA and CUG. Column is complete. Next double C, CCU, CCU, CCC, CCA, CCU. No, CCG is last. CCG. Next CA, CAU, CAA. Next CAC. Sorry, upper is C, no? Upper is C. Here is A. Next CAG. Next CGU, CGC, CGA and CGG. So second column is also over. Next third column AUU, AUC, AUA and then AUG. Next AU, not AU, sorry, AC it is. ACU, ACC, ACA, ACG. Next column this one, double A, AAU, AAC, AAA and AAG. And next AG, AGU, AGC, AGA and AGG. Got. Next last column GU, GUU, GUC, GUA and GUG. Over. Next column GC, GCU, GCC, GCA and GCG. And last, next one, not last, last one, GAU, GAC, GAA, GAG. And now is the last one, that is GG, double G, GGU, GGC, GGA and GGG. See, so these are the different type of the codons we got. Totally, see here, 64 codons are there. Each column containing how much? 4 and 16 columns are there. 16 into 4 means totally 64 codons are there. And all these 64 codon now what used for what? Naming the amino acid. Naming amino acid. Totally how many type of amino acids are there? 20 amino acids are there. Then you can ask here 64 codons are there ಅಂತ ಕೇಳೋದು. ಹೌದು 20 amino acids ಇದಾವೆ. ಆದರೆ ಒಂದು what amino acid ಗೆ ಒಂದಕ್ಕಿಂತ ಜಾಸ್ತಿ ಕೋಡ್ಸ್ ಇದಾವೆ. ಇಲ್ಲಿ ಗೊತ್ತಾಗ್ತದೆ ನಿಮಗೆ. ಒಂದು ಅಮಿನೋ ಆಸಿಡ್ಗೆ ಒಂದಕ್ಕಿಂತ ಜಾಸ್ತಿ ಕೋಡ್ಸ್ ಇದಾವೆ. ಅದು ಹೇಗೆ ಅಂತ ಕೇಳಿದ್ರೆ ನಿಮಗೆ ಗೊತ್ತಾಗ್ತದೆ. ಸಿ and it is here, it is given in the book also it is given. given. See, this is PHE. PHE andre the phenylalanine. PHE ge enanta kartala phenylalanine, and next group is called as leucine. LEU, it is a leucine. Then so this entire group is also leucine. Idu kuda leucine. LEU andre the leucine. You can also write equally require. Then ಇಲ್ಲಿ 3 group is that AUU, ACE and AUA, and it is isoleucine. ಇದಕ್ಕೆ isoleucine ಅಂತ ಕರೀತಾರೆ. And then next all 3 ಇಲ್ಲಿ all 3 value ಇದೆಲ್ಲ value VIL ಅಂತ ಬರ್ತದೆ. ಇದರಲ್ಲಿ the GUG AUG ಮಾತ್ರ MET, AUG ಮಾತ್ರ MET ಮಿಥಾಯೋಲಿನ್. ನೆಕ್ಸ್ಟ್ ಬರ್ತದೆ ಅದೇ ಏನಂತ ಹೇಳ್ತಾನೆ. ನೆಕ್ಸ್ಟ್ this entire group is serine, SER, and then next group is proline, PRO, it is PRO, and next one is threonine, PHR, and next one is alanine, ALA, next one is alanine, ALA, and then next two is a tyrosine, TYR, and these two are stop codon, stop, and first two is HIS, histamine, HIS, and next gilliman, GIN, and next two is ASN, adenosine, and next one is a lysine, LYS, next one is a lysine, next two is ASP, aspartic acid, and the next two is a glutamine, GLU, and then next CYS, cysteine, CYS, and then this is again stop, and then next UGG is a tryptamine, TRP, and next four all are the arginine, and next one is a serine, SER, and next one is a arginine again, arginine, and then last one is glycine. So that means here all the 20 amino acids are named. Ella 20 amino acids anna name madidave. Ella 20 amino acids alli nodi, the first one is phenylalanine, PHE. Okay, next one is what? The leucine, and that is LEU. You can write it in the bracket, LEU. So next third one is isoleucine, ILEU, isoleucine. Next fourth one is what? Methionine, MET. Next fifth one is valine, VAL. Next sixth one is the serine, SER. Next seventh one is the proline, PRO. Next eighth one is threonine, THR, and next ninth one is alanine, ALA. Next 10th tyrosine, TYR. Next 11th one is histamine, HIS. Next gilliman, GIN. Next adenosine, ASN. Next lysine, LYS. Okay, next one A aspartic acid, ASP. Next one, glutamine or glutamic acid, GLU. Next, cysteine, CYS. Next, 19, tryptamine, it is a TRP. Next, sorry, it is a number change, 15, it is 16, 17, 18, 19. Tryptamine again, serine. Serine is more, I think. Yes, over. Next, arginine. ARG. And last, 20. Glycine. GL1. See, these are the 20 different amino acids, 20 different type of amino acids, and these all amino acids here have more than what, the one or two the codes, and because of this reason here the genetic code has given some characteristic features. Genetic codes ಗೆ ಕೆಲವು characteristic features ಇದಾವೆ, and let us start with the first one by one. The genetic codes ಗೆ ಏನು characteristic features ಅಂತ ಕೇಳಿದ್ರೆ, so first one, genetic codes are initiator. First character. What is that genetic code as initiator? So genetic codes are initiator ಅಂತ ಹೇಳಿದ್ರೆ, during the protein synthesis always one code that help in initiation of the protein synthesis. MRNA ಮೇಲೆ ಆ ಫಸ್ಟ್ ಕೋಡ್ ಇರೋದೇ ಇನಿಷಿಯೇಟರ್ ಕೋಡಾನ್, ಅದು ಪ್ರಮೋಟ್ ಮಾಡಲಿಕ್ಕಿರೋದು, and that codon is called what here, the AUG. AUG is what, initiator codon, and that codes for amino acid called as methionine. That codes for amino acid called as what, methionine. So that is why AUG is the initiator codon. AUG ಇದ್ರೆ ಮಾತ್ರ protein synthesis ಆಗ್ತದೆ, ಇಲ್ಲ ಅಂದ್ರೆ ಆಗೋದಿಲ್ಲ. ಅದಕ್ಕಾಗಿ the code AUG is called as initiator codon, and it codes for what amino acid? Methionine amino acid only. Okay, next, the next one is the codes are the universal. Codes are universal. Next one, what is this codes are universal? So codes are universal means here from prokaryote to the eukaryote, multicellular to the tissue grade, organ grade, etc. system, here a particular order, the code having a particular amino acid only. Particular code ಗೆ particular amino acid only ಇರ್ತದೆ. ಅಂದರೆ triple UUU codes for phenylalanine only, not for a serine, not for a proline or anything. Got it? ಅಂದರೆ ಟ್ರಿಪಲ್ ಯು ಇದ್ರೆ ಅದು ಫಿನಾಲನಿಗೆ ಮಾತ್ರ, ಇಲ್ಲ ಟ್ರಿಪಲ್ ಯು ಇದ್ದು ಸಿ ಇದ್ರೆ ಅದು ಫಿನಾಲನಿಗೆ ಮಾತ್ರ. ಹಾಗಂತ ನೆಕ್ಸ್ಟ್ ನೋಡಿ, ಡಬಲ್ ಎರಡು ಯು ಇದ್ದು ಒಂದು ಎ ಇದೆ, ಆವಾಗ ಫಿನಾಲನಿಗೆ ಕೋಡ್ ಆಗೋದಿಲ್ಲ. ಸಿ ಇದ್ರೆ ಮಾತ್ರ ಫಿನಾಲನಿಗೆ ಕೋಡ್ ಆಗೋದು. ಯು ಎ codes for leucine. Similarly, see here the CUU also code for leucine, and then CUG also code for leucine. So means the one amino acid having more than 2, 3, 4 what here codes are there. So that is why here a particular codon having a particular amino acid only. For example, you see CGU codes only for arginine, CGU only codes for arginine, not for a tryptamine or not for a serine. That is one universality. Next one, the codons are degenerate. Codons are degenerate. Here the codons are degenerate andre, codons are degenerate means here what, the one amino acid having more than 2, 3 what codes. ಒಂದೇ ಒಂದು ಅಮಿನೋ ಆಸಿಡ್ಗೆ ಒಂದಕ್ಕಿಂತ ಅಥವಾ ಎರಡಕ್ಕಿಂತ ಜಾಸ್ತಿ ಕೋಡ್ಸ್ ಇದ್ದಾವೆ ನೋಡಿ. ಈಗ ಲ್ಯೂಸಿನಿಗೆ ಎಷ್ಟಿದ್ದಾವೆ? 1, 2, 3, 4, 5, 6 ಲ್ಯೂಸಿನಿಗೆ. ಸಿಮಿಲರ್ಲಿ ವ್ಯಾಲಿನಿಗೆ ಎಷ್ಟಿದೆ ನೋಡಿ? 1, 2, 3, 4. Alanine how many are there? 1, 2, 3, 4. And that is called a degenerate. See, glycine you see 1, 2, 3, 4. Arginine also having what? 6. So like this, one amino acid having more than 2 what here codes are there, and that is what you call as what? degenerate. Next, then codons are commaless. Codons are commaless. ಏನಿದು commaless ಅಂದ್ರೆ ಒಂದ್ಸಲ ನೋಡಿ ನಾವು mRNA strand ಅನ್ನು ಓದಲಿಕ್ಕೆ ಸ್ಟಾರ್ಟ್ ಮಾಡಿದ್ರೆ if I start to read the mRNA and that is AUG CCU and AUC CUA AAA CCA. So these are the 5' end to what? The 3' end mRNA strand, and on the mRNA strand only what is depicted? Genetic codes are depicted, and genetic codes are always read in how many, what the sequence? The 3. So while you are reading, AUG is initiated codon first starting irrelevant. AUG. So like this you have to read as a set of 3, like this AUG, CCU, then next AUC, then CUA, like this 3 sets ಅನ್ನು ಮಾಡ್ತಾ ಇರಬೇಕು. ಅಲ್ಲಿ ಎಲ್ಲಿಯೂ ಕಾಮಾ ಬರೋದಿಲ್ಲ. Don't put any punctuation mark like comma, then what this comma, ಹೀಗೆ ಕಾಮಾ ಬರೋದಿಲ್ಲ. No punctuation mark. ಏನೋ punctuation mark ಹಾಕಬಾರದು. ಕಾಮಾ ಇಲ್ಲ, full stop ಇಲ್ಲ, exclamatory sign ಯಾವುದೂ ಇರೋದಿಲ್ಲ. And that has to be read from 5' end to 3' end, not from the 3' end to the 5' end, 5' end to the 3' end. As a set of 3, that is a triplet codon. It is called as a genetic code and without any punctuation mark, and that is what you call as what? Commaless. Next one, the genetic codes are terminated. Genetic codes are terminated. ಏನಿದೆ terminator ಅಂದ್ರೆ genetic codes are terminated ಅಂತ ಹೇಳಿದ್ರೆ on what here the mRNA strand, ಇಲ್ಲಿ mRNA strand ಅಲ್ಲಿ at the end of the what the protein synthesis ಸ್ಟಾಪ್ ಆಗಲಿಕ್ಕೆ ಬೇಕು. ಸ್ಟಾಪ್ ಆಗಬೇಕಂದ್ರೆ ಇಲ್ಲಿ ಸ್ಟಾಪ್ ಕೋಡನ್ ಅಂತ ಇದೆ ನೋಡಿ, UAA, UAG and these, and these 3 codons are UAA, UAG specific, ಅದು ಚೇಂಜ್ ಆಗೋದಿಲ್ಲ ಹೆಸರು, UAA, UAG and what UGA. These are the 3 codon. They do not code for any amino acid, remember this, and that is why they are called as a stop codon. Another name is called as what? A terminator codon, or they are also called as a nonsense codon. Nonsense ಅಂದ್ರೆ ಅದಕ್ಕೆ ಏನಿಲ್ಲ, ಸೆನ್ಸ್ ಇಲ್ಲ, ಯಾವುದೇ ಅಮಿನೋ ಆಸಿಡ್ಗೆ ಅದು ಕೋಡ್ ಮಾಡೋದಿಲ್ಲ. ಒಂದು ವೇಳೆ ಮಧ್ಯದಲ್ಲಿ ಎಲ್ಲಿಯಾದರೂ ಈ ಸ್ಟಾಪ್ ಕೋಡಾನ್ ಬಂತು, UA ಬಂತು, UAG ಬಂತು ಅಥವಾ UGA ಯಾವುದು ಬಂತು ಅಂತಂದ್ರೆ ಪ್ರೋಟೀನ್ ಸಿಂಥೆಸಿಸ್ ಅಲ್ಲಿಗೆ ಸ್ಟಾಪ್ ಆಗಲಿಕ್ಕೆ ಬೇಕು. ಯಾವುದಕ್ಕೂ ಒಂದು ದ ಎಂಡ್ ಅಂತ ಇದ್ದೇ ಇದೆಯಲ್ವಾ, ಹಾಗೆ ಸ್ಟಾಪ್ ಆಗಲಿಕ್ಕೆ ಬೇಕು. ಈ ಮೂರರಲ್ಲಿ ಯಾವುದಾದ್ರೂ ಒಂದು ಇದ್ದೇ ಇರ್ತದೆ. in a long chain mRNA ಅಲ್ಲಿಗೆ ಸಿಂಥೆಸಿಸ್ ಸ್ಟಾಪ್ ಆ್ಯಂಡ್ ವಾಟ್ ದ ಟರ್ಮಿನೇಷನ್ ಟೇಕನ್. ಟರ್ಮಿನೇಷನ್ ಅಂದರೆ ಸಪ್ರೇಟ್ ಆಗಿಬಿಡ್ತದೆ, ಪ್ರೋಟೀನ್ ಮಾಲಿಕ್ಯೂಲ್ ಫಾರ್ಮ್, ಒಂದು ಪ್ರೋಟೀನ್ ಮಾಲಿಕ್ಯೂಲ್ ತಯಾರಾಯಿತು, ಆವಾಗ ಸಿಂಥೆಸಿಸ್ ಸ್ಟಾಪ್ ಆಗ್ತದೆ. ಮತ್ತು ಪುನಃ ಬರಬೇಕಾದ್ರೆ AUG ಬರಲೇಬೇಕು ಆ ಸ್ಟ್ಯಾಂಡಲ್ಲಿ. ಸೊ ಹಾಗಾಗಿ ದ ಜೆನೆಟಿಕ್ ಕೋಡ್ಸ್ ಆರ್ ವಾಟ್ ದ ಟರ್ಮಿನೇಟರ್. ಸೊ ಟರ್ಮಿನೇಟರ್ ಜೆನೆಟಿಕ್ ಕೋಡ್ಸ್ ಆರ್ ದ. ಆ್ಯಂಡ್ ದೆನ್ ನೆಕ್ಸ್ಟ್ ಒನ್ ಈಸ್ ಹಿಯರ್ ದೀಸ್ ಕೋಡಾನ್ಸ್ ಆರ್ ಸೆನ್ಸಿಬಲ್ ಆ್ಯಂಡ್ ಅನಾಂಬಿಗಸ್. Codons are sensible and unambiguous. Andre during the protein synthesis, here what any the amino acid not code with the wrong what codon. ಯಾವುದೇ ಒಂದು ರಾಂಗ್ ಕೋಡಾನ್ ಜೊತೆಗೆ ಅದು ಏನಾಗೋದಿಲ್ಲ, ಮಿಂಗಲ್ ಆಗೋದಿಲ್ಲ until it bring proper what the anticodon. Until it what bring proper anticodon. ರಾಂಗ್ anticodon ಜೊತೆಗೆ ಯಾವಾಗಲೂ ಅದು compromises ಆಗೋದಿಲ್ಲ, and that is the characteristic is called as what here sensible. And the last one is codons are non-overlapping. Codons are non-overlapping. Easy ಏನಿಲ್ಲ, non-overlapping ಅಂದರೆ ನಾವು ಇದನ್ನು ಓದಬೇಕಾಗಿದ್ರೆ ಸೆಟ್ ಆಫ್ 3 ಅಂತ ನಮ್ಮ ಮೊದಲನೇ ಹೇಳ್ತಾ AUG CCU ಹೀಗೆ ಮಾರ್ಕ್ ಮಾಡಬಾರ್ದು, ಅಂಡರ್ಲೈನ್ ಕೂಡ ಮಾಡ್ಬೋದು. ನಾನು ಇದನ್ನು ಅಂಡರ್ಲೈನ್ ಮಾಡಿ ತೋರಿಸ್ತಾ ಇದ್ರೆ ಯಾಕೆ ಕೇಳಿದ್ರೆ ನೀವು ಓದಬೇಕಾದ್ರೆ AUG ಅಂತ ಓದಿ ನಂತರ ಮತ್ತೆ ಪುನಃ ಅಂತ ಹೋಗೋದು GCC ಅಂತ ತಗೊಳ್ಳೋದು ನೀವು, ಅಂದರೆ ಓವರ್ಲ್ಯಾಪ್ ಆಯ್ತಲ್ವಾ, ಇಲ್ಲಿ ನೋಡಿ ಓವರ್ಲ್ಯಾಪ್. ಸೊ ಲೈಕ್ ದಿಸ್ AUG CCC AUC ಹೀಗೆ ಓದಬೇಕಾಗಿದ್ರೆ ಇದು ಸೆಟ್ ಆಫ್ 3, AUG ಒಂದು ಸೆಟ್, CC ಒಂದು ಸೆಟ್, AUC ಒಂದು ಸೆಟ್. ನೀವು ಓದಬೇಕಾಗಿದ್ರೆ AUG ಓದಾದ್ಮೇಲೆ ಮತ್ತೆ ಪುನಃ ಯು ಕೆನಾಟ್ ರೀಡ್ ದ GCC ಲೈಕ್ ದಿಸ್ or CA like this. It is not correct, and that is why codons are non-overlapping. So these are the 7 important characteristic feature of the genetic code. Idu genetic code the important characteristic feature. I hope that you understand about this. So with this basic idea, now we have to go for the next what is the process and that is the what here the translation. ಇನ್ನು ಮುಂದೆ ಬರೋದೇ ನಿಮಗೆ ಟ್ರಾನ್ಸ್ಕ್ರಿಪ್ಷನ್ ಆಲ್ರೆಡಿ ಯು ಆರ್ ಸ್ಟಡಿಡ್. ಈಗ ಬಟ್ ಆಫ್ಟರ್ ದ ಟ್ರಾನ್ಸ್ಕ್ರಿಪ್ಷನ್ MRNA ಸ್ಟ್ಯಾಂಡಲ್ಲಿರುವಂಥ ಜೀನ್ಸ್ ಕೋಡ್ ಅಂದರೆ ಜೀನ್ ಅಂದರೆ ಜೆನೆಟಿಕ್ ಕೋಡ್ ಅದು ಕೋಡ್ ವರ್ಡದಲ್ಲಿದೆ. ಅದನ್ನು ಓದೋದು ಹೇಗೆ, ಅದು ರೀಡ್ ಮಾಡಲಿಕ್ಕೆ ವಾಟ್ ಆ್ಯಂಡ್ ವಾಟ್ ಈಸ್ ರಿಕ್ವೈರ್ಡ್, ಪರ್ಟಿಕ್ಯುಲರ್ಲಿ ದ ರೋಲ್ ಆಫ್ ದ ಟ್ರಾನ್ಸ್ಫರ್ RNA is very important. Transfer RNA is also produced by what? DNA, and that process is called a transcription, and the transcription transfer RNA, and that having a two end, that is one is anticodon binding end and another is what the what amino acid binding end. ಹಾಗಾಗಿ ಅದು DNA ದಿಂದ ಹೊರಗೆ ಬಂದು DNA ದಿಂದ ಅಂದರೆ ನ್ಯೂಕ್ಲಿಯಸ್ ದಿಂದ ಹೊರಗೆ ಬಂದು ನಾವು what the translation process ಆಗಬೇಕು, and how exactly translation process etc take place you have to study, and that is about the genetic the code. Now next one is genetic code. Next one is according to your syllabus genetic code and mutation. Mutation and the genetic code. Mutation and genetic code. Mutation and the genetic code. What is a mutation, you know? So mutation means sudden change in the what DNA, sudden change at the what gene, and so that is what you call as a mutation. So many different type of mutation is there. Alteration of chromosome, alteration of DNA, alteration of entire gene, or alteration of sequence of the gene, sequence of the DNA. That also you studied the point mutation. Now you give the example for the point mutation. No, remember already you studied in this class also, and YouTube class also you studied the sickle cell anemia is the best example for the point mutation. Sickle cell anemia, point mutation, in which the only one sequence, one word, one alphabet change agide. Now, and because of that one alphabet, here the sixth position of the beta globin, what changes occur, and because of this, here what the mutation taken place in them, and thereby here wrong hemoglobin instead of normal, the S hemoglobin formation taken place, and that S hemoglobin make problem to the what human being. So ಹಾಗಾಗಿ ಇಲ್ಲಿ ಜೆನೆಟಿಕ್ ಕೋಡ್ ಎಷ್ಟು ಇಂಪಾರ್ಟೆಂಟ್ ಅನ್ನೋದನ್ನ ಇಲ್ಲಿ ಎಕ್ಸಾಂಪಲ್ ಸಮೇತ ಹೇಳಿದ್ದಾರೆ ನೋಡಿ. ಸೊ one example we can take, see here Ram has in a triplet code ಅಂತ ಕೊಟ್ಟಿದ್ದಾರೆ. Ram has a red cap. ಸೊ ಜೆನೆಟಿಕ್ ಕೋಡ್ ಅಲ್ಲಿ ಹೇಗೆ ಚೇಂಜ್ ಆಗಬಹುದು? ಸಿ Ram has a red cap ಅಂತ ಹೇಳಿದ್ರೆ ಇಲ್ಲಿ what the deletion or addition, ಡಿಲೀಷನ್ ಆಗ್ಬೋದು, ಅಡಿಷನ್ ಆಗ್ಬೋದು, ಡಿಲೀಷನ್ ಆಗ್ಬೋದು, ಎಡಿಷನ್ ಆಗ್ಬೋದು, ಅದು ನಾವು ಹೇಗೆ ಬೇಕಾದ್ರೂ ಮಾಡ್ಕೊಳ್ಬೋದು ಇಲ್ಲಿ. ಆ್ಯಂಡ್ ಸೊ ದಟ್ ಕ್ಯಾನ್ ಬಿ ಹಿಯರ್ ವಿ ವಾಟ್ ಸ್ಟಡಿ ಬೈ ಒನ್ ಎಕ್ಸಾಂಪಲ್. ಸಿ ನೋಡಿ ಈಗ ಏನಾಗ್ತದೆ ನೋಡಿ, ಈಗ ಈ ಜೆನೆಟಿಕ್ ಕೋಡ್ದಲ್ಲಿ ಅಡಿಷನ್ ಅಂತ ಮಾಡ್ಕೊಂಡು ಇಲ್ಲಿ ರಾಮ್ RAM HAS RED CAP ಎಲ್ಲ ಟ್ರಿಪ್ಲೆಟ್ ಕೋಡಲ್ಲಿದ್ದಾವಲ್ಲ, ಕರೆಕ್ಟ್, ಎಲ್ಲವೂ ಟ್ರಿಪ್ಲೆಟ್ ಕೋಡಲ್ಲಿದ್ದಾವೆ. ಈಗ ಎಕ್ಸಾಂಪಲ್ ನಾನು ಹೇಗೆ ಬರ್ತೇನೆ ಕೇಳಿದ್ರೆ ಇದಕ್ಕೆ ಅಡಿಷನ್ ಮಾಡ್ತಾ ಹೋಗ್ತೀವಿ ನಾವು. ಒಂದೊಂದು ಲೆಟರ್ ಅಡಿಷನ್ ಆಗಿದ್ರೆ ಏನು ಚೇಂಜ್ ಆಗ್ತಾ ಹೋಗ್ತದೆ? ರಾಮ್ ಹ್ಯಾಸ್, ಕರೆಕ್ಟ್, and then I add B, BREDCA and P. P ಒಂದು ಸಪ್ರೇಟ್ ಆಯ್ತು, ಮತ್ತು ಮುಂದೆ ಇದೆ, ಯು ಡೋಂಟ್ ವರಿ ಅಬೌಟ್ ದಟ್. ಓಕೆ, ಸೊ ಹಾಗೆ RAM B ಒಂದು ಅಡಿಷನ್ ಇದೆ, ಯಾವುದರ ಮುಂದೆ R ಮುಂದೆ B ಒಂದು ಅಡಿಷನ್ ಆಗಿದೆ. ದೆನ್ ಒನ್ಸ್ ಅಗೇನ್ I add I, BIREDCAP, ಎರಡು ಲೆಟರ್ ಬಂತೀವಿ. ದೆನ್ ಒನ್ ಮೋರ್ I add RAM HAS BIG, G ಎಕ್ಸ್ಟ್ರಾ ಮಾಡಿದೆ, ಬಿಗ್ ಆಯಿತು, ಆ್ಯಂಡ್ ದ್ಯಾಟ್ ಇಟ್ ಬಿಕಮ್ ರೆಡ್ CAP. ಹಾಗಾಗಿ ಈಗ ಈ ರೆಡ್ ಮುಂದೆ ಯಾವ್ದು ಎಕ್ಸ್ಟ್ರಾ ಕ್ಯಾರೆಕ್ಟರ್ ಬಂದ್ಬಿಡ್ತಾಡೋ